So we move to our next Caparica talk, which is going to be given by Dr. David Nicolau, who I personally acknowledge for being here today. Well, David Nicolau comes from the United States, of course, from the PCCP, from the Center of Anti-Infective Research and Development in the Hartford Hospital. David is the pharma director, and he's going to talk to us about redefining carbapenem resistance. Thank you very much for being here today.
Well, good morning. That was your attempt, my attempt at audience participation. You know, when I too Googled this talk as titled by my colleague, it says that it's supposed to be inspirational, unlike a typical business talk, a series of slides with no opportunity for interaction. So the success of my presentation is almost entirely dependent on the audience. And so I need your help, I need your opportunity to embrace the process. We're talking about issues related to gram-negative infections. And when we talk about this, you know, oftentimes we talk about a specific patient that provides inspiration. But as presented by Karen and our colleague from McMaster, when you look at issues related to gram-negatives, you know, if you work in hospital, you work in laboratory, you work in development, or perhaps you've had a family member in hospital, it's very likely that these gram-negatives have wreaked havoc with their lengths of stay, or perhaps impacted morbidity and mortality. And we very cavalierly talk about many of these terminologies and mechanisms of resistance. We heard that the WHO, you know, arguably an informative group of colleagues, put in place some of the critical challenges in gram-negative infections. They noted the challenges of Acinetobacter, Pseudomonas aeruginosa, and last but not least, the Enterobacteriaceae, and summed up the challenges as being carbapenem resistant. And so the terminology that we use quite frequently, this terminology of CRE, I think it lacks informativeness in a very, very big way. And in the next few minutes, I want you to think about what a term like CRE means. What's in the name? It is not a single entity.
It's a normal miraculous.
That's correct. That's correct. See, this is the kind of enthusiasm that we want during these sessions. Dr. Livermore is already inspired. How good is that to get David inspired? It took but yet a little pinch and a little tweak. But we want you to understand that you need to be a little bit deeper in the dive. CRE is no longer an applicable term to either the development of new medicines or the management of patients. And I want to show you why that might be the case. This is an unfortunate patient. We see many of them that have multiple comorbid conditions. They've multiple times, in this case, for multiple urinary tract infections, and the patient has now failed and come to hospital. He's gone directly to the emergency room, and from the emergency room to the ICU. The organism that presents itself several days after is a Klebsiella pneumoniae, but it carries with it this terminology of MDR. And the laboratory reports to us that it is multi-drug resistant, but also resistant to the carbapenems. And so that would be what people would view as CRE. But look at the flavor, the phenotypic profile of this organism. It has resistance as defined in the laboratory to ertapenem, but as a meropenem susceptibility. This should stop and make you ask questions about how this should be defined clinically and what the implications are for how you might manage this patient. So I often use slides that ask questions of the audience. And many of you are in hospital, many of you are clinicians. And so the question is, how would this organism be reported in your hospital setting? Would it be reported as a carbapenem-resistant enteric? Would it be reported as producing enteric? Or would it be reported as a non-carbapenemase, carbapenem-resistant enteric? Think about that for a moment. What would generally happen? In most laboratories, this would be reported as CRE.
No, because I would beat them up for using the term CRE.
Thank you.
It's a shocking
Give it to me. Give it to me. Right? So this is absolutely true, but I think you would agree that in the majority of the laboratories, this would be out as CRE. That is, it's woefully inadequate relative to the diagnosticians.
The original Proteus, as I said,
Correct.
Carbapenem resistant.
That is correct.
Wasn't CRE.
That is correct. Absolutely. And that's an intrinsic mechanism of resistance. So we've tried to use a variety of phenotypic profiling in the laboratory. I'm part of the CLSI for very good or perhaps for very bad at the current moment relative to our discussions with global harmonization. And we've tried to use various phenotypic profiles, albeit imperfect. Some of them are a little bit quicker. The modified carbapenem inactivation tests, a little bit more sensitivity and specificity relative to the carbapenemases. All of these testing methodologies suffer from one important fact in the context of what we heard from Dr. Bush this morning, and that is the development of targeted, very specific beta-lactamase inhibitors. These phenotypic profiling tests don't tell you what the carbapenemase is. And that's the major challenge with this approach in use in clinic. We have increasingly relied on multiple platforms for the detection of these beta-lactamase genes. We happen to land and we use the Cepheid system at my hospital. We use the Carba-R testing system. And we do that because we have the platform in place. We use it for staph, we use it for TB, we use it for lots of things, including the carbapenemases. And what these molecular diagnostics tests will tell you, they'll probe for the gene. And they'll tell you that the enzyme or the gene is there. But what it also doesn't address, it doesn't address the hydrolytic nature and the functionality of these genes, which is clearly important, as Dr. Bush has said. But many of these tests, and it's true here, we're talking about very rapid results. And that's important in the context of the clinic, because not only could you choose that precision medicine, the right beta-lactamase inhibitor, but you can do it relatively quickly and get patients started on a good medicine. If you think about these Enterobacteriaceae, generally the most prevalent of organisms in both hospital and community-based infections, and we've generally lumped these CREs into one bucket. But it is very much two buckets. You have the non-carbapenemase producers, that patient I showed you, with that very strange carbapenem phenotypic profile, resistant, meropenem susceptible. And if you look historically, these organisms would tend to be small groups of organisms, low percentages. As we now start to probe these carbapenem-resistant Enterobacteriaceae, in many parts of the world, these non-carbapenemase producers have come up as fairly large populations, surprisingly to many. And then you have the population of organisms that often get most of the attention. And perhaps they should, because as Karen said, they denigrate the beta-lactam backbone and make beta-lactams, including carbapenemases, essentially useless for the management of serious infection. And within those carbapenemase producers, there are multiple flavors. We talk about the big five, KPC, OXA, NDM, VIM, and IMP, but there are also many other flavors that are yet unappreciated. But if you're going precision medicine and you're going to drive efficacy, you need to be able to differentiate the flavors of CRE and then differentiate the enzymology of CRE. And so if you look at the left side of this, the non-carbapenemase producers, and I acknowledge my colleague for sharing this slide with me, you see mutations causing outer membrane impermeability, porins, modifications, and losses with some enzyme-mediated resistance, AMP-C or ESBL. This is what drove the phenotypic profile in that initial patient. There are some important factors, though, that we need to recognize for potential clinical care. One is ultimately treatment. Two is infection control. These types of resistances are not on plasmids, so they're not shared quite easily among organisms. That makes infection control people much happier, right? Times for response and how you manage those patients. And the medicines we choose might be, in fact, very different. If you look at a data set from my state in Connecticut in 2017, and you look at all the CREs that were dumped over to the state laboratory, when they actually went through them and asked the question, how many were carbapenemase producers? Only 1/3 of the CRE were carbapenemase producers. 2/3 were non-carbapenemase producers. And if you look at the non-carbapenemase producers here, what you see is that many of them are going to be carbapenem susceptible to meropenem. And this provides some indication, perhaps, with the right dosing of something like meropenem, although it's carbapenem resistant by the clinical laboratory, we might be able to drive clinical efficacy. And while many of you don't have all of the new BLBIs, beta-lactam, beta-lactamase inhibitors, you have meropenem. And so it's really important to understand what's driving resistance mechanistically. But don't forget, with all of this new and very sexy technology, we still want to do the classic phenotypic testing, automated susceptibility testing. We still want to know what the MIC values are, because that's going to help us differentiate how best to optimize therapy. If you look at the CP, CREs, the carbapenemase producers, again, the vast majority, as one might expect, we're in North America, are KPC, a little NDM, a little OXA, and a little IMI showing up in that population. And you can certainly see for the carbapenemase producing CREs, most of these antibiotics are quite terrible. We have colistin here, my least favorite antibiotic for a variety of reasons, not something that we use unless we absolutely have to. And then we have ceftazidime avibactam, the newest and the initial beta-lactam lactamase inhibitor as approved, and has quite great potency for carbapenemase producers, as well, interestingly enough, as non-carbapenemase producers. And why is that? Dr. Bush told us. She said that many of these enzymes are either cephalosporinases or they're serine-based enzymes with the non-carbapenemase producers. Avibactam, a great inhibitor of those enzymes. So it puts the package together what can be done. Data are needed, you know, in the context of managing patients with non-carbapenemase producing CRE, some data from our colleagues in Baltimore suggesting that the aggressive doses of carbapenems that we developed, 2 grams Q8 of meropenem with a prolonged infusion, might actually be quite management. But we need additional clinical data to support those decisions. And then you have the carbapenemase producing side of the equation. These are clearly challenging enzymes. Tremendously high levels of resistance. The values aren't just above the breakpoints for the carbapenems. They're 512, 1024. Something that with pharmacodynamic optimization of dosing, you still can't touch. We need alternatives. We need the new beta-lactam, beta-lactamase inhibitors. And again, importantly here, concern not only from a patient individual perspective, but certainly from an epidemiologic and an infection control perspective, we're talking about these resistances being on plasmids, and they can move very, very quickly. So rapid dissemination. If you look historically at what we've done, as I said, admittedly, I don't have an extra hour to say enough bad things about colistin, but it is an antibiotic that we brought back from the doldrums, simply for one reason, because we had very little with respect to antibiotics that had activity against, notably, CREs, Pseudomonas, and Acinetobacter that are resistant to carbapenems. The problem with colistin, in short, though, is that it's difficult to give enough drug to be clinically effective and not overtly toxicity. That's the challenge with colistin. So I think some of the discordance between the in vitro discovery and the clinic is those translational medicines and what we can deliver safely to patients. So what have we done? Dr. Livermore alluded to this. We add things like meropenem and tigecycline and try to make drugs that are not so adequate a little bit better. But it's challenging. So we've moved to doing a variety of things. One is understanding on the carbapenemase side, what are the enzymes that are driving those resistances? Again, KPC, OXA48-like, it's important to realize that this encompasses a variety of different enzymes, 232 and 181. There's a variety of different enzymes that are in here falling into the OXA48. And then the metallo beta-lactamase enzymes. Again, we have some different things coming. I think much of the metallo beta-lactamase that we see for resistance is an artifact of the way we do in vitro testing, and it has very little to do with what happens in the clinic, but you'll see some of that data at upcoming meetings from our group. And when you start talking about understanding the enzymology, and then you have new medicines that have preferential activity against specific enzymes, Ceftazidime avibactam, KPC and OXA, meropenem avibactam, KPC, imipenem rel, and others, you can begin to start designing regimens, right, combined with molecular diagnostics that are going to give you the right opportunity to drive things very quickly. I included this slide simply because it makes the point that these new BLBIs, they have much better pharmacokinetics, right? They have less variability. They're easily tested in the laboratory. S equals success. If you're giving these agents and you're giving them in sufficient doses for various degrees of renal function, you can drive efficacy. And so these are non-randomized trials with ceftazidime avibactam, where the agent is generally used as salvage therapy. And you can look at the efficacy of these papers. It's remarkable. Reduction in infection-related morbidity, mortality, a reduction in colistin-related toxicity. So these are important additions, but opportunities to drive efficacy. Again, I don't want you in the clinical laboratory to abandon susceptibility testing. We've asked a lot of our colleagues on the diagnostic side, and not only are we asking them to give us the enzymology, but we want them to be able to rapidly differentiate multiple flavors of these KPC-based isolates. And that, so I'm told by the diagnostic folks, is a real challenge. This just tells you that KPC comes in a bunch of flavors. Karen Bush already told you that. But the implications are KPC2 versus KPC3 and a variant. This is going to have a very different phenotypic profile, right? KPC3 variant might see higher MICs to ceftazidime avibactam. And it may be one of the reasons why we have multiple ELBIs on our formulary. We don't want one size fits all because they're going to have different potencies. based on the variants within those organism types or enzymology. So it's quite important that we understand all of the aspects. So you can see that CRE as a term is not a very encompassing term. We need to ask very different questions. And last but not least, Dr. Bush mentioned this with the Canadian data, the concept of now having co-producing metallos and serine-based carbapenemases. We get referrals for these patients with great frequency now. And so the genotypic profile is NDM and OXA, plus a little bit of an ESBL in there to keep it interesting. And you can see that this organism is highly resistant to everything, including colistin. And so, you know, the molecular diagnostics help give us some idea, but many don't have current molecular diagnostics in their laboratories. And so what we have been doing is using an old school approach, using the strip diffusion methodologies, the E-test or the Lifo-Philchem test, and you lay this strip down with a gradient of antibiotics from low to high. Ceftazidime, no zone. Aztreonam, no zone. But when you put them together, you begin to see something develop here. And we term this development the zone of, because it goes from having no antibiotic activity to one that with the combination of antibiotics makes biological sense, right? Ceftazidime AV is inhibiting the KPC and inhibiting the ESBL. It allows aztreonam to be aztreonam for the metallo-beta-lactamase. And so we've now looked at this because we're getting reports and we're getting organisms sent to us quite frequently that are very much co-producers of serine and metallo-beta-lactamases. So it's something that can be done in the laboratory very quickly. And it's by no means perfect. And there's a bunch of variability. And it's certainly not standardized. But it may inform therapeutic options that could be useful for the management of your patient. And it's something that can be done in the clinical laboratory. Very little resources. And we think it's important to give clinicians information that helps them make good choices about how to manage their patients with critical illness. You heard a lot about this over the previous talks, the fact that we have lots of different enzymes, and importantly, the hydrolytic capacity of these enzymes can be very, very different. And laboratory molecular-based diagnostics are not going to tell you this. So again, I want to stress the fact that we still continue to need MIC values. If you're running the clinical laboratory, don't abandon them. We need to be testing multiple agents. We can do some PK/PD and optimization. Issues related to fitness are of interest to us. And last but not least, I think as we have more whole genome sequencing, as we have a better sense, we're going to have an idea of what the clonality is of many of these organisms. And I think this is the next missing part. You put a wimpy enzyme in a bad clone, it does bad things. Put really virulent, very fit enzymes, hydrolytic enzymes on bad clones, and it does bad things to many, many people, right? So we certainly want to have an idea about what that is, and we're going in that direction. And then lastly, you know, the overall piece of phenotypic profile. You know, if that's the resource you have, and you don't have molecular diagnostics, then you use it. But what you have to recognize is there are certainly some shortcomings. And most importantly is the fact that it doesn't differentiate the enzyme. And if you're just using something, polymyxin plus meropenem plus tigecycline, not knowing the enzyme, maybe that's okay, because it's a very broad regimen that isn't based on enzymology. But if you're getting into using the BLBIs and you want to optimize the quality and efficiency of care, our colleagues said we don't use these new medicines because they're expensive. I tell people the most expensive antibiotic is the one that doesn't work, right? An expense relative to what? Day in the hospital, day in the ICU, morbidity, mortality. so we can drive some of that efficacy. Genotypic profiling, the opportunity for precision medicine. This is great stuff because as clinicians, we can define how best to optimize those regimens. It isn't just throwing things on the wall. It's making specific regimens based on the enzymology. We have the ability to do that. Differing enzymology, different therapy, rapid results. 48 minutes for the platform that we use after the organism is taken and put into the system. But you have to realize that not all clinically relevant enzymes are being identified currently on these readily available molecular platforms. So it's not the end-all be-all. We very much like this gradient diffusion strip. We think the zone of hope does just that. It provides some opportunity to inform clinicians, especially as we see multiple co-producing enzymes and places in the world where they don't have access to the molecular diagnostics. And last but not least, the presence or absence of carbapenemases. The need not to use CRE as a term, but to use non-carbapenemase producing, carbapenem resistant, or to use carbapenemase producing carbapenem resistant organisms has implications not only for the quality of care and decisions about how you manage those patients with respect to choices of medicine, but importantly, it has implications for infection control and saving not only the patient, but actually, conserving the potential for resistance in your hospital. Thank you very much.
Thank you very much, David. Excellent talk. Thank you. So we have time for questions. If you have, yep. Karen.
I fully appreciate the difference between CRE and CPE. However, from a pharmaceutical company's perspective, CRE, if you're selling a carbapenem, you want to know, is it going to be susceptible or is this organism susceptible or resistant? A clinician wants to know that first. Then you drill down into your CPEs or non-CPEs. But I think that CRE still has some validity and some meaning for people who are working in drug discovery.
Yeah. I think certainly we can use some of this terminology, but I would hope that on the clinical front, we're becoming more informed. And when it comes to managing the patient and optimizing the care, the terminology CRE has very little applicability now. Matter of fact, it might in some cases, Karen, we've seen, it actually drives people in the wrong decisions. They don't quite understand what it means. It also means that if you have CRE, but it's not a carbapenemase producer, maybe you're using some of these broader spectrum BLBIs that may not be necessary. And as pointed out in the conference, one of the concerns with using them is the inherent development of resistance and inappropriateness. So again, I think the better we can differentiate what's a producer, what's not, gives us an opportunity to drive efficacy and the appropriateness, stewardship, in the context of therapy.
I'm not denying that. I'm simply saying that if you're developing a drug, that chart that I showed that said % resistance, that's what matters to a pharmaceutical company.
Right.
And they start with that, and then they ask why.
Correct. But one of the connections and the challenges is you see some of these companies fail, or you see some of these situations where drugs are not being used. Part of that is because you develop it in the CRE bucket, but the actual use and the stewardship and the practicality of it is developed in the non-carbapenemase producer and the carbapenemase producer buckets. Those are the challenges in the context of commercialization.
Can I join in on this?
Please.
But surely one sees this writ large with the travails of plasmamycin, which is an aminoglycoside touted as an anti-CRE drug. Now, needless to say, there is no carbapenemase, nor ever will be a carbapenemase that attacks plasmamycin. It's completely different. The issue is, well, has the organism got an aminoglycoside modifying enzyme, in which case plasmamycin might be quite good at avoiding it, or has it got a methyltransferase? So talking of plasmamycin activity against CRE leads us actually into a lot of potholes.
Yeah.
So I don't think that pharmaceutical companies help themselves at all in talking of activity of non-carbapenems against CRE, because it's other mechanisms that are pertinent.
Let's close.
But I think CRE is being used as a marker for multi-drug resistance.
Yeah.
And rightly or
Right.
wrongly,
Right.
when we look at some of these lists from the CDC and WHO, unfortunately, we're not seeing multi-drug resistant Acinetobacter.
Yeah.
But it's a lot easier.
We can follow during the coffee break because we are really delayed. Thank you very much. Thank you very much, David, for this nice talk.